The following is reviewing the understanding as it relates to Unit 2 AP Biology curriculum. Please note the disclaimers associated with this video as well before we begin. This is reviewing Unit 2, Tonicity. Specifically, let's go back and review what osmosis is. When we think of osmosis, we think of water. So whenever you think of osmosis, you're thinking of water. But we don't need to talk about water today. We're talking about the understanding of water relative to cells. So osmosis in general is diffusion of H2O across any membrane. So think like a wall. How does it go back and forth? And that is what we call the term osmosis. Okay. So oftentimes we're talking about whether or not there's a higher concentration of water on one side relative to another when we talk about osmosis in general. Now, you might have seen things like just a side note, if you're talking about AP biology, oftentimes they give a question, and the question usually has something like a U-tube, not to be associated with YouTube, but a U-shaped tube. And they usually have some sort of barrier, and you have to try to figure out what is happening in the concentration of that barrier as it relates to a solute and a solvent. So that's the other high yield topic you're expected to understand. If you click the link below on my link tree, you can go to one of my stores and click on Unit 2, and there are Unit 2 related FRQs and multiple choice questions that have a ton of problems related to YouTubes. However, for today, what we're reviewing is not only just osmosis, but the topic of tonicity. T-O-N, oh boy, I need to check how to spell that word. Tonicity, my wonderful spelling. So Tonicity, which is related to osmosis relative to the barrier as it's associated with a cell, and our understanding of that behavior relative from one side of the cell to the other side of the cell as it is associated with the barrier of a cell wall or a cell membrane. So that is what we're going to be reviewing today. Now, in this video, you will remember that the animal cells are up top and the plant cells are on the bottom. And in this picture, you see these water molecules, these wonderful, wonderful water molecules all around the picture and our little arrows. And today we're going to have to figure out what is happening with the water molecules. Are they going in? Are they coming out? What really is going on when we talk about those water molecules? We have to go ahead and figure that out. So we're going to try to figure that out by first reviewing what is happening in the differentiation between what's normal and what's not normal. So when we take a look at the first one that we're going to view, we're going to take a look at the center relationship. We now know that we have animal cells and we have plant cells, but how water is viewed is different depending on the environment they're in and what happens to the cell. Remember, animal cells have a cell membrane and plant cells have a cell wall in addition. So that's the item that makes them different. Now, in general, for both plant and animal cells, if we have these really cool water molecules coming in and coming out, that means that the environment is going to be called isotonic. So that means that there's equal concentrations of water coming in and coming out, and that's both for plant and animal cell. However, when we think about what's normal, what's not normal, here lies the conundrum. What we consider normal for a plant cell is not going to be the same as a animal cell. For example, when we're taking a look at an animal cell, for example, humans, and it is in an isotonic environment, we consider that to be normal. So that is considered normal. So we like that. So we're just going to put a little check mark next to the normality of that cell in an isotonic solution where the water concentrations are equal on both sides. However, for a plant cell, remember, there is that cell wall. So it's not going to like the equal concentrations of water going in and out. So there's a bunch of excess here. We don't like it. And what ends up happening is if we clear this out, clear all drawings, and we go to the next slide, we can see that it leads to what is called flaccid. So flaccid is a unique word. Flaccid just basically means limp. So if you ever take a look at a plant, and you see that sometimes you might think, okay, well, if I water my plant every day, it will be okay. And what ends up happening is sometimes if you have a plant and you water it and you have equal concentrations of water going in and out, then like this would be my plant before, but sometimes afterwards it will limp. So tilt over. So that's what the word flaccid means. So to review, in both isotonic environments for plant and animal cells, there's equal concentrations of water going in and out. However, it is not the same environment that we choose to decide to have it in, because in animal cells, this would be a perfect condition, but for plant cells, it would be flaccid, which would mean that it will tilt or limp. It would be limp. So let's take a look at this area right here. So as you can see, water is going into our cell and we are constantly just pushing in that water. And when we're pushing in that water, what is happening is we are now in what's called a hypo, which I should write down here. We are in a hypotonic environment. So, or hypotonic solution, I should say. So we're hypotonic for both of these, which basically means that there is less solute or salt concentration than inside the cell. So on the outside than inside the cell. So what happens is water is just going to enter nonstop. So you have water that's constantly flowing in. So if you think of an animal cell, what ends up happening is if you have too much water, and what's going to end up happening is it's going to lyse. It's going to lyse. And this doesn't really do a great picture of it, but you could see like a little thing right here, it bursted. That's what lyse means. We basically are bursting. So the cell will eventually swell up, fill with water, and then burst. So water enters for an animal cell. Now, for a plant cell, the response is different. Water enters, and then the cell becomes what's called turgid. Turgid. And it remains intact because of the rigid cell wall, which we like. So we like that rigid cell wall to be there because it provides us an opportunity to ensure that water enters in and there is more water inside than outside. So that gives it a turgid environment, which allows it to be normal for a plant cell. So in a hypotonic solution, just to review, if you have a animal cell, it will burst or lyse. If you have a hypotonic solution with a plant cell, it's normal and will be called turgid. And a way that you can think about it is hypo, like if you're hyper, think of a child that is hypertonic, they oftentimes are consuming a lot of sugar. Think of like that. But if you have a hypotonic solution, you're not going to consume a lot of sugar. So you're just sort of still and meaning you're not consuming a lot of solute compared to your outside environment. So you can think of sugar being a solute, which it is, and saying that, okay, if I'm inside, if I consume a lot of sugar, then I'm going to be hypertonic. But if I don't, in this case, I have more water going in, I'm considered hypotonic. So that leads to the final scenario, which is this last column right here, which is going to be our hypertonic solution. So Hypertonic for both plant and animal cell. And this is when our water molecule right here is leaving. Our water molecule is leaving out. And what ends up happening is if there's more water that's leaving out, we end up shriveling. So you can think of cells really shrinking because there's no water going inside, or there's water constantly leaving, especially to compensate for that hypertonic solution where it's super salty on the outside. So it wants to compensate for that. If you have a plant cell, you're going to have what's called plasmolyst. So plasmolyst is a unique term which basically means the same thing. Water exits, but the plasma membrane will pull away from the cell wall. And this could be detrimental. So this would be not good. So this would be really bad, not normal. We don't want this. In this case, plasmolysis for plant cells would be not normal. So turgid would be normal, but plasmolysis is not normal. And so those are the three types of different ways that you can take a look at the way animal cells are versus plant cells. If you want a more, feel free to screenshot this photo or keep this in your notes, but this is the differences between tonicity for the three different environments because it tells you the difference between hypertonic and hypotonic and isotonic solution. But I think this picture right here really sums it up really well to better understand the differences and what is normal and what's not normal in animal and plant cells.